To keep everything on track, I'm Linnaea Ostroff from the University of Connecticut. I'm here with Joshua Vogelstein and Sarah Bradley, and we welcome you to the third NeuroNex meeting. We want to thank the NSF for sponsoring and all our wonderful speakers and panelists and session chairs. We're very excited for this meeting. The talks are going to be pre-recorded. What you can do during or after the talks, if you have questions, is please post them to the chat, and the session chair will monitor the chat and invite you to ask your question live, or maybe read your question for the speakers or panelists to answer in the Q&A session afterwards. This is going to be on YouTube Live. It is also being recorded. The diversity discussions are the only part that will not be recorded and will not be live streamed, but we'll talk about that later. There are going to be breakout rooms available if you want to go off and have smaller conversations. I'd like to remind you that we have a code of conduct. Please, if you haven't already, take a look at that. It's linked from your email and from the agenda. And please, of course, remember to treat each other with respect and kindness, and not only because it's being streamed live. So Joshua, do you want to make any further points?
I do, thank you. So I would like to add that our theme is the dimensions of team science diversity, and so we'll be having a lot of discussions about that in various ways. And there will be a poll, a Google poll link from the chat. Is completely voluntary, but we encourage everyone to participate to help discuss these ideas. And we want to encourage especially trainees to ask questions. This, I think, is probably the biggest NeuroNex meeting ever, partially because NeuroNex is much larger now, also because NSF granted everybody in the world availability to join this, which is an amazing thing. So of the almost 400 attendees, I think nearly 300 are funded by NeuroNex, and another 100 people are here to learn about NeuroNex and are not part of the program. And so we welcome them and everybody to ask questions, as Linnaea said, using the chat. And I think that's it. Linnaea, did I miss anything?
The poll that we're going to ask you to fill out is absolutely anonymous. There'll be a link put in the chat. I think Sarah will do that soon. This poll will be open all day today up until tomorrow morning, and the results are going to be presented tomorrow afternoon. Again, this is voluntary and it is anonymous, but we encourage you to fill it out, and you'll get more information about these sessions a little later.
And so with that, I think we can hand it over to NSF NeuroNex program directors.
I'm Reed Beaman. I'm a program director at NSF in the Directorate Division of Biological Infrastructure. I'm one of the program directors that manages the NeuroNex program.
Good morning, all. I'm Claire Hemingway. I'm in the Office of International Science and Engineering, and delighted to have the chance to partake in bits of this. I'm connecting through my role with being contact for Canada and just the international component.
Okay, I'm Sridhar Raghavachari. I'm also a program director and managing the NeuroNex program, as well as in the neural systems cluster in the biological sciences directorate. I know it's a mouthful.
So I'm Floh Thiels. I'm just like Sri. I'm a NeuroNex like Sri, Reed, and Claire, everyone who preceded and who will follow. I'm one of the NeuroNex program directors, and also in the Directorate for Biological Sciences, Integrative Organismal Systems. And I wanted to mention that Christoph Limbach, who is a program director from the German Deutsche Forschungsgemeinschaft, DFG, as well as Charlotte Inchley, who is a program director from MRC UKRI, all NeuroNex program directors, as well as Maxime Baudouin from the Fonds de Recherche Québec, are at this meeting. They're not here right now, but they are also part of the program, NeuroNex program directors present. So reach out to the NeuroNex program directors if you have questions.
Good morning. I'm Bridget Turaga, a program director in NSF's international office, along with Claire. And this is my first NeuroNex investigator meeting, so I'm quite pleased to be here. Thank you.
And with that, we are handing it over to Joanne, and welcome Joanne Tornow. She's the Assistant Director of the Directorate for Biological Sciences. Thank you very much, Joanne, for joining us. And I forgot to mention also, thank you very much to Linnaea, Joshua, and Sarah for organizing this really largest NeuroNex investigator meeting. Very exciting.
Well, thank you, Floh, for the handoff. And thanks to everybody who's in the Zoom room and watching on YouTube for joining us for the third annual NeuroNex investigator meeting. Likewise, I am quite excited by the engagement, the level of interest, and the potential that'll come out of this gathering. And I also would like to thank the meeting organizers, Joshua, Linnaea, the meeting coordinator, Sarah, for your very hard work in putting all this together. It is a non-trivial task to coordinate across multiple international partners in a virtual environment. So congratulations. So over the next 2 days, you'll all be discussing topics at the forward edge of neuroscience, neurotechnologies, and approaches for advancing our understanding of the brain. Not a insubstantial topic. You'll be discussing you're also going to be talking about a novel method of conducting research in the field of neuroscience, one that's really at the core of the NeuroNex program, team-based inter and transdisciplinary research. As described in the theme of this meeting, Dimensions of Team Science Diversity, the teams that make up NeuroNex networks leverage diversity of experience by engaging scientists across the globe, each with a unique background and approach to solve grand challenges in neuroscience. I'm glad to see that each panel at this meeting involves a representative funded by NSF's international partners. It really helps to embody the global coordination and collaboration at the heart of the program. As we at NSF are working to integrate biology more broadly, we're building upon this team-based model, which is a bold shift from individual investigator paradigms. Examples include our new biology integration institutes and our recently announced solicitation for AI institutes, which this year includes an AI to advance biology theme that may be of interest to you. The goal of this new solicitation is to connect AI researchers with biologists, in this case neuroscientists, to develop new AI designs that help us better understand the relationship between neural circuits and behavior. And broadly speaking, these integration efforts connect researchers across disciplines and backgrounds and build on recent advances across and outside of the biological subdisciplines. They promise to provide more than the sum of the complementary expertise and knowledge brought by the individuals involved, producing new technologies, data, and insights that will advance our understanding of the world around us. We believe the discoveries, technologies, and approaches that emerge from your efforts will have implications for other fields within the biological sciences and across the sciences and engineering broadly. In the end, the exchange across sciences will hopefully result in applications with societal impact, such as robots that can smell disease or advanced brain imaging technologies. Of course, while team science has its benefits and presents a number of opportunities, no method or approach is without its obstacles, as will be discussed during this afternoon's keynote roundtable. For example, team science requires a new way of thinking to identify efficiencies built into networks, discussion of and planning for workflow practices and management, and new methods of communication. All of these challenges are heightened as NeuroNex teams become larger and more geographically dispersed with the recent Phase 2 awards. We must also work to incentivize collaborative experimentation, data sharing, and analysis to bring the power of convergent approaches from multiple disciplines to neuroscience. And in addition to these challenges to efficiently conducting team science, there's cultural obstacles in academia and elsewhere that could inhibit this method from becoming more prevalent. And I do want to note here something that I think all of you already know. Team science does not equate to big science. Bio and NSF value greatly the contributions of individual investigators. With an activity such as NeuroNex, we're enabling individuals from diverse disciplines to work collectively toward a big fundamental question and thereby amplifying your individual contributions. To address the need for additional collaboration and data sharing, the most recent awards under the NeuroNex program will contribute to the development of and comply with data sharing and standardization best practices and policies. The teams, representing 70 investigators, nearly half of whom are affiliated with a research organization not based in the US, will produce data that will be accessible to the community and enable further research and spur new investigations. For example, there's a new network examining how primates maintain information for brief periods of time, which will create new single-cell gene expression data that will be invaluable to the broader scientific community. And this recent round of awards builds upon the advancements and technologies that were created through the networks and hubs funded in phase 1 of the program and through other investments that have been made as part of the BRAIN initiative. The network, led by researchers at the University of Texas, for example, will make use of advances in 3D electron microscopy, made by investigators supported by phase 1 awards. And the From Odor to Action network has its origins in groups originally formed as part of an NSF Brain Initiative Ideas Lab. In addition, carbon fiber electrode arrays developed using previous NeuroNex funding will help the communication, coordination, and control in neuromechanical systems network study the mechanisms that underlie motor output in response to and interaction with an animal's environment. Sorry, that was a lot of words all in one phrase. My hope is that as these new networks build on advances, you will build on the discussions you have today and tomorrow by continuing to talk about them and the ideas you have when you return to your labs, your institutions, your networks, and your research. The new data and tools you learn about will no doubt be of use, but the ideas about the method by which science is conducted and who conducts that research, including examining individual dimensions of race, gender, discipline, and scientific approach, as well as what types of institutions are engaged in research, are of great value as well. Building on the discussions that occur here, or the virtual version of here, when you return home, and having similar discussions at your institutions and in your labs, especially with your trainees, will bring fresh ideas and perspectives to the field of neuroscience and lead to new methods of training and education that will benefit not only neuroscience, but biology and the sciences and engineering more broadly. By sparking those new ideas, developing new methods of training the next generation of the STEM workforce in this team science approach, and by creating a more connected and diverse field, we will be able to develop a better understanding of how the brain works and produces behavior, which in turn could revolutionize everything from medicine to artificial intelligence. And with that hope, I will once again welcome you to this important meeting, and I thank you for being here. And I turn it back to you, Floh.
Thank you very much. That was great. I would like to switch over now to Sam Howerton, who also wanted to join the welcome remarks.
Thank you very much. It's always hard to follow Joanne because her and her team are so exhaustive in terms of their capture of what's going on. I just want to take a few moments and talk about how NeuroNex, I think, fits within the larger context of international collaboration and why it's so important, really, for us as the U.S. science community. I think it goes without saying that we all realize that the events that we find ourselves in at the moment has impacted mobility, has impacted our ability to cooperate with one another simply because of the barriers to travel. But that hasn't prevented us and groups like you from cooperating with one another virtually to continue to move forward the frontiers of science, which is critical to NSF's success, not only here domestically, but internationally as well. And so with partners like Canada and Germany and other major economies, we think that it's really important for the investigators such as you to continue to promulgate the message that the United States is committed to international collaboration, committed to discovery, both today and in the future. And so I will leave with that there because I know we are running out of time, but I want to welcome you all as well as the same way that Joanne did. And thank you once again for not only all of the work that you've done, but all the work that you will do to ensure that we continue to support this great, great series of investigations. Thank you.
Thank you very much, Sam. And with that, I hand it over to, I believe, Kristen, unless Joshua and Linnea have an intermediate step before we move to the first session.
Okay, well, I too want to thank Linnea and Josh and Sarah and all of the going to really be, I think, an outstanding set of sessions today and tomorrow. And I'm really looking forward to hearing them all. You should note that all of these were pre-recorded, and I notice in my own talk that there's going to be an error in what I say about when Tom Bartol's going to be speaking, because it looks like he's been moved to today's schedule. There may be a few other things like that the pre-recording will have a slightly different agenda order because they were prepared before the final agenda was produced. But what I'd like to encourage people today is that while these talks are going on, please put your questions, and especially trainees, I really want to encourage you, the NSF and the organizers in particular want to see participation by our trainees and our junior participants through the Q&A. So please put your questions into the chat room. We're going to go through all of the talks, the pre-recorded talks first, but you can just keep putting your questions in as the talks go. And then later this morning, we will have a session of Q&A. That will go through those questions that are in the Zoom chat room. And I'd like to encourage you to, you don't have to turn off your face during the talks. I presume we've got plenty of bandwidth. But for sure, during the Q&A, I'd like everyone to keep your video on so we have a real feeling of discussion and not just talking to names and avatars. So the more that we can have our faces showing, the more lively this meeting will be. So at this point, I really would like to turn off my remarks and ask Kelly to queue up the talks. And please be engaged and put your questions into the chat room, and we will come back to them in about 30 minutes. Thank you, Linnea and Jovo, for organizing this workshop. Today, I'm just introducing our two grants within this, the NeuroNex teams. We have a 2017 NeuroNex 1 hub entitled Enhanced Resolution for 3DM Analysis of Synapses Across Brain Regions and Taxa. And we're designing instrumentation to improve 3D-EM and collect information in greater detail and use high-performance computing to increase the rate of knowledge discovery. And I have three co-PIs on this grant, two co-PIs rather, James Carson and Terry Sanowski. And Tom Bartol out of Terry's lab will speak later tomorrow in session 4 about one of our major tools that we've completed, nearly completed production of. We also have a brand new grant, NeuroNex3, enabling identification and impact of synaptic weight in functional networks. And we have 4 teams, and I'll show you the individual teams momentarily. So our overarching question is, what constitutes synaptic weight, and what role does it play in shaping neural circuits, and how does it change during growth and plasticity? And we're going to focus this first year on mitochondria as our primary topic of interest to focus on their role in each of these components. IRG1 is a molecular. It's led by Silvio Rizzoli and has Aaron Schumann, Radu, Alice, Volker, Stefan, Paul, and Ruben. And Silvio will give the talk later today. IRG2 is focused on subcellular constituents. And I am the lead overall PI, but Mark Ellisman is our team lead in IRG2. Brian Jones, Eric Jorgensen, and Niels Bros are all participants in this IRG. And Christian Rosenman will be speaking today about the topics that he's contributing to this IRG. IRG3 is looking at ultrastructural motifs of synapses actually in connectomes and neural circuits with defined states. This team is led by Clay Reed at the Allen Brain Institute, and we have Greg Jeffress, Moritz Helmstetter, Nariman Kasturi, and Linnea Ostroff as part of this team. And tomorrow, or later today, Davi Bock, in fact later this morning, will be speaking about his contributions in this IRG. IRG4 is to produce cyber infrastructure to enable the multi-scale discovery of this synaptic weight in neural circuits. This is led by Terry Sanowski. Uri Manor will be speaking this morning. Josh Vogelstein, Flavi, James, and Viren Jain from Google are all part of this infrastructure. And here are the 4 talks that will be given, or the 5 talks. Tom Bartle tomorrow, Silvio Rizzoli, Christian Rosenmund, Davi Bock, and Uri Manor today. Not quite in that order, but they're listed here by their IRGs. And we also have many team members who are giving poster presentations. So I hope that you will attend their posters to learn about the details. In particular, John Mendenhall's poster will show you more about our tomography on the scanning electron microscopy. And this is a tool that's being under development and further development within both of our IRGs.
What we're doing in IRG3 is essentially attempting to place fine-scale ultrastructural detail within the context of larger-scale circuit reconstructions, or connectomes. The large-scale work that we're doing, this is an example of it. This is largely manual tracing done in a whole fly brain data set acquired by my lab and collaborators and released a few years ago. And this is about a 750 micron wide and 350 micron tall and 250 micron deep EM image volume with tremendous complexity. And the wiring that you're seeing here is just a subset of that. To understand what's in there better, in addition to these quote-unquote wiring diagrams, we'd really like to look at the detail. And we're fortunate in that this data set has attracted a fair amount of attention from the segmentation community. So, for example, Jan Funke at Genelia Research Campus and colleagues have done a complete synaptic segmentation of this data set, and this is an example of that work. And then Peter Lee and colleagues at Google have done a segmentation of the neurites, as well as the Flywire project at Princeton by Sebastian Sung. And we used that segmentation to accelerate reconstruction of a circuit we were interested in the fly mushroom body. And this is showing that work, so, you know, over 1,000 Kenyan cells reconstructed. And we can do pretty powerful analyses of those connectomes. In this case, I won't go into details, but we were able to see order, whereas previously this circuit had thought to be random. But we would really like to understand how the details of synapses fit and ultrastructural details fit within those connectomes, and how those two classes of information can complement each other. So to give you a sense of what I mean, here we see an overview of the entire EM volume, one section of it. And if we just zoom in, we see fantastic resolution is available within the data set. And here we're zooming in on a single synapse. And this is really, you know, oversampling the resolution of the data set. But what we can do, if we like, is take a picture of that same spot in the data set and see the detail. And here we see synaptic vesicles and presynaptic specialization called the T-bar and the cleft and the postsynaptic objects. And so what we'd like to do is annotate these structures and look at our connectomes with this kind of detailed information in mind, you know, cleft size and so on. Of course, there's yet more detail. This is tomography from a collaboration with Mark Ellisman, who's also in the NeuroNex collaboration. And here you can see lipid bilayers. And so what we'd like to do is upsample our data in collaboration with Uri Manor, and then segment it and get all of this information out as much as we can at this very high level of detail. While we wait for our collaborators to do that, we can really get a nice head start. We can take publicly available segmentations like the synapses I showed you earlier, or forthcoming work from the Fly EM team at Genelia. They have a different data set subset of the whole brain, but it has mitochondria segmented, and we can look at connectivity as a function of proximity to mitochondria. And we can do that same thing in mammalian data. So this is just a taste of what we're planning to do in IRG3, but hopefully it gives a sense. Thank you.
Hi, everyone. My name is Uri Manor. I'm honored to be a member of the IRG4 group that's led by Terry Sinowski at the Salk Institute. And what we will be doing is using advanced computational methods to make sense out of all the data that all the other groups are going to be generating. And what I'll be talking about today is just one small part of that we're calling deep learning-based super resolution microscopy. And this is just to illustrate why you would want to do that. On the left and on the right is the exact same sample being imaged with the exact same pixel dwell time on a scanning electron microscope. But on the left, we're using 8 nanometer pixels, and on the right, we're using 2 nanometer pixels, which means that on the left, we can image 16 times faster than the one on the right, which is great. But as you can see, there's not as much resolution, there's not as much detail, it's much noisier, so we can't pick out all of the subcellular structures that we're going to be interested in, such as presynaptic vesicles. While on the right, we can see it, but just to make the point about how long this takes, on the right, if we wanted to image the brain with the resolution on the right, it would take 160 years, whereas on the left, it would just take 10 years. So we have a vested interest in being able to image faster. So long story short, without going into too much detail, we developed a deep learning model that allows us to take images like what you see on the left, which are very low resolution. Again, you can't see the vesicles. And then we run it through our deep learning model. And on the middle, you can see what the output is, where we have a nice, crisp image where you can have much more resolution, much more contrast, much more signal to noise. And on the right is, just for reference, our ground truth, so-called ground truth data that we took in our own microscope. So one of the really nice things about our model is that it actually works for other people's data that was acquired on other microscopes, so out of the box. So that's really important because we're going to be working with data from many people, from many labs, and the fact that our model is so robust and can be used out of the box for multiple data sets is really nice. So it means that we can use it right away. So on the left and right, you can see an example of the input versus output from a FigSem data set from Janelia Farms. And one thing I'll point out is that because this data is so much more crisp, we've removed the noise, we've increased the resolution, this data is much easier to segment, which is a really important part of the analysis pipeline that we'll be working on. So I imagine our model will be plugged into the whole segmentation pipeline, which is super important. And I'll also quickly point out that this method also works for live fluorescence data. So here you can see some neurons that we're imaging live mitochondria, and we're watching them traffic up and down. So using this deep learning method, we can now get much better resolution and then be able to image with much higher speed. So we can look at things like mitochondrial dynamics in a live cell. And as a next step, one of the many small things that we're working on is correlative microscopy, where we do live videos, and then we fix the cell, and then we do EM. And then we have an EM image to overlay with our live videos. And we're going to build a classifier, a deep learning-based classifier, that can predict whether a mitochondria was moving or not, and it learned from these videos like these that we're taking. And then we can apply that to all of our EM data, so then we can have dynamic information about the mitochondria that we're looking at in our electron microscopy data. So with that, I just want to thank, there's way too many people, I don't even have time to say all of the names, but this was a massive effort, big collaboration. I also want to thank some of these sources of funding, including, of course, the NeuroNex. And with that, I'll take any questions and suggestions. Thank you.
Hello, my name is Silvio Rizzoli, and I would like to speak about our NeuroNex2 project, which aims to understand synaptic weight in functional neuronal networks. I am part of IRG1, which is going to find the molecular determinants, the characteristic markers for defined synapse states, like silent states, like potentiated states, and so on. IRG1 is composed of 8 scientists from the UK, Canada, Germany, and the US, and works mostly at the nanoscale, relying on both culture neurons and brain preparations. For example, we investigate the structure of individual molecular complexes or the structure of full synapses using cryo-EM. We also rely on innovative labeling protocols, which allow us to see molecules at both the electron microscopy and fluorescence microscopy levels. We also use complex and highly precise protocols to understand synaptic turnover and protein secretion. And we integrate different methods to visualize synapse proteomes, such as electron microscopy and quantitative biochemistry. Finally, we use live imaging to define synaptic states, and we then study the ultrastructure of the different synapses to understand the potential markers of these states. To give you a couple of examples, I switched to some pretty movies of synapses in action. You now see the movement of different soluble molecules in the presynaptic compartment. You see these gray clouds, which are synaptic vesicles. On these timescales, they are not very mobile, and they are surrounded by 28 types of different soluble molecules, which are moving, and they give you an idea of how motion in the presynapse would actually look like. On the next slide, you're seeing a movie of the postsynaptic compartment, and this is a postsynapse under normal activity conditions, in which you see about 450,000 different proteins shown in the right copy numbers and in the right positions, obtained from a combination of quantitative biochemistry, super resolution imaging, and electron microscopy. The challenge that we now face is to take such movies, which are extremely quantitative, to take them and instead of just having them from a situation like normal activity, to have such quantitative information in such movies now for different synaptic states, to understand with very high degree of accuracy the behavior of different synaptic states. This is going to be a huge challenge, but we hope to be able to face it. And when we are done with this project, we are now going to understand how different synaptic states are characterized and what the markers for these states are. Thank you for your attention, and I look forward to your questions.
My name is Kristen Rosemund. I'm from the Charité Medical School in Berlin, Germany. I'm happy to present you the RG2 consortium, which consists of 6 PIs. And we deal with the question of subcellular constituents and their role in manifesting defined synaptic states. So I would like to highlight a project that I do together with Niels Bose from Göttingen, which deals with the question how we can measure synaptic strengths in electron microscopy. So the hypothesis is based on that synaptic strength is predominantly determined by the content and the topology of synaptic transducers. For example, where are calcium channels positioned relative to fusion-competent vesicles, how many and where are the AMPA and MDA receptors are positioned across the synapse. So the aim is to develop an EM-compatible toolset that allows us to count and map these key synaptic transducers at the synapse. So for this, we have identified adding his tags to the extracellular domain of these synaptic transducers as a useful approach. You can see here some example staining of a calcium channel in the synaptic cleft. And with this tool in hand, we have did some proof of principle experiments. For example, we measured the distance between fusion-competent vesicles and calcium channels. You can see here that these calcium channels in this particular synapse are very close to the vesicles, approximately within a radius of a synaptic vesicle. We can further use this to do functional anatomy using flash and freeze technology pioneered by Erik Jorgensen's group. So by comparing the distribution of vesicles before and after action potentials, we can see which vesicles have preferentially fused after an action potential. You can see here that that vesicles approximately 30 nanometers away from a calcium channel preferentially fuse. So with having these feasibility experiments been done in cultures, we would like to make these tools now available for the entire consortium by developing knock-in mice that express these tags in 4 key synaptic proteins. We'd like to test their labeling feasibility in 3D tissue and subsequently quantify putative changes in architecture of the synapses in different plasticity states in 2 model hippocampal synapses. We think our project also links very nicely with the first part of our consortium, which deals with architecture and dynamics of pre- and postsynaptic machineries, and also have strong links to advanced microscopy technologies developed in the Jorgensen and Rizzoli lab. Thank you.
I'd like to thank the organizers for giving me this opportunity to introduce to you Alinum Swift, which is a cross-platform Python GUI for the signal whitening Fourier transform image registration toolkit created by Art Wetzel. And then the GUI was created by myself, Bob Kaczewski, and a Python API from Daniel Wagoner. Alinum Swift was motivated by the need that quality 3D EM demands quality alignment. It was developed as an aim of the NIH BTRR at mmbios.org, and it has been integrated and deployed as an aim of NSF NeuroNex3 at 3DEM.org. You can access it at the 3DEM workbench. There's a wiki page that shows exactly what I'm going to show you today, and a Git repo at that link. I will now demonstrate for you live Alinum Swift. So we begin by loading a stack of images, which I've already done, saving it as a project, setting a hierarchy of scales for the images at full scale, 1/6 scale, and 1/24 scale. Generating those scales, which I've already done, switching to the coarsest scale, setting up for an initial estimate of the affine transform, and then saying align all. It runs on 48 CPUs of a Skylake node at Stampede 2 at the Texas Advanced Computing Center. And it takes about 25 seconds to align at scale 24. During this time, the images are being aligned pairwise with each other. The init affine takes 19 seconds to estimate, and then now it's generating the aligned images by applying the transform to the base images. There's the alignment. I can now switch to scale 6, do a refinement of that initial alignment, and we're going to do successive approximation or improvement of the alignment. I say align all at scale 6. These images are 4K by 4K in size. It takes about 35 seconds to do the alignment at this scale. We are also projecting the images onto a bounding rectangle big enough to contain the offsets of all the alignment. And also we're removing the average value of the offsets so that the images are nicely centered within the bounding rectangle image. And when it completes, we can see that alignment. At this point, I would go to scale 1 and do the alignment there, which takes about 9 minutes, but I've already performed that step. And we can see that by switching back to the presentation. And here is a movie of the alignment. On the left is the full-scale image resized to fit on my window, and then a small square window of a enlarged area that is shown on the right-hand side. And there you can see the quality of the alignment, which is pretty darn good. And in future versions of the software, we will add elastic affine transform to the image. I'd now like to acknowledge the collaborators in the Harris Lab who contributed all the images and input on the design of the GUI and workflow. James Carson at TAC, who set up the 3dem.org workbench. Art Wetzel, Bob, Terry Sinowski, my boss at Salk, and Daniel Wagoner at Caltech, and of course our funding agencies, NIH and NSF. Thank you.
Hello, my name is Bobby Kasturi. I'm an assistant professor at the University of Chicago and a neuroscientist at Argonne National Labs. I'm here to present for just a few minutes about the work we are doing and we did for our first NeuroNex grant. In the next couple of slides, I'll just explain to you the work that we're doing and why we think it's important. So basically, the goal of our first NeuroNex grant was to develop a pipeline that would allow us to image entire brains, entire mouse brains, 1 centimeter cube, at arbitrary resolutions, i.e. we would like to image the same mouse brain either at the resolution where we can see brain regions, or at the resolution where we can see individual neurons or their processes, and then ultimately at the highest resolution where we can actually see how neurons connect with each other. The way we decided to approach this problem was to use a microscopic and sample prep imaging that works on any species and ultimately works with a combination of synchrotron X-rays and, from other work that we've done, automated serial electron microscopy, which is the workflow that you see here. One thing that's important to mention on this slide is that basically the staining works with heavy metals and ultimately plastic embedding of samples. And it's important again to say that this works on all kinds of species. One of our goals for this NeuroNex grant was not only to optimize this for the mouse, as the data I'll show you, but also for other organisms like the octopus, the squid, the butterfly, etc. So rather than belabor it, I thought I would just jump and show you exactly what the data looks like. So here, for example, is work that we did for this effort where we imaged an entire mouse brain, something that is 1 centimeter cubed at something like 1 micron resolution. And even though this is a downsampled movie that I'm showing you, probably you can already tell that even at this resolution, you can make out individual myelinated tracts, large brain regions, and because of the myelinated tracts, how they communicate with each other. But of course, this isn't the full resolution of the data set that we have. What we actually have is this entire brain at the resolution you're going to see in the next movie. So here, we've zoomed into this hippocampus, one part of the brain. This is closer to the full resolution of our data set. Each of these little spheres, circles that you see appear and disappear is a neuron in the hippocampus. There's white streams that are flowing out are the dendrites of those neurons. And these little black squiggles are the individual myelinated axons. So if you can imagine, we have that entire brain done at this resolution. And one of the reasons we're interested in doing this is we would, for the first time, like to get accurate counts and accurate measures of where every neuron and every brain region is relative to each other, so that it could serve as an unbiased way to compare across multiple species. Thank you very much for your time, and I look forward to your questions.
Hi everyone, my name is Vikram Chandrasekhar and I'm a graduate student at Johns Hopkins University working in Joshua Vogelstein's lab. Today I'm going to be talking about CloudReg, a tool we developed for automated cross-modal brain volume registration in the cloud. As many of you may know, mice are among the most commonly studied animal models for understanding brain physiology and pathology. In fact, the BICCN has a goal to have a complete cell atlas of the mouse brain by 2025. With the advent of clearing methods like Clarity and iDisco, neuroscientists can generate whole, intact brains significantly faster and at a higher resolution than previously possible. Here we have 2 examples of light sheet imaged Clarity samples. On the top, we have a small data set that can be as big as 20 gigabytes, and on the bottom, we have a large data set that can be as big as many terabytes. So cleared tissue presents unique artifacts in both of these cases, like uneven illumination, for example. And these kinds of challenges require scalable tools for image processing and registration. To address these needs, we developed the CloudReg pipeline. CloudReg starts with high-resolution multi-field of view raw data, performs an initial intensity correction step, and then stitches these data together into a complete volume. We then perform an additional intensity correction so that subsequent registration is more accurate. We then register these data to the Allen Reference Atlas, and here you can see the Allen Reference Atlas labels overlaid on our sample. And these data are then uploaded to cloud storage for visualization with a web-based interactive viewer like NeuroPlancer. We then transform these data to the Allen Reference Atlas for comparison with other samples and subsequent analysis. Here is an example of a NeuroPlancer link that you can view in your browser. And here is a link to our code so you can deploy this pipeline for your samples as well. Here are some additional results that we have on Clarity samples. Here we have a low resolution and high resolution Clarity sample, raw data on the left, and the same samples transformed to the Allen Atlas on the right-hand side. In order to test the generalizability of our pipeline, we also registered human MRI samples, macaque MRI samples, and rat clarity samples, as you can see in the results here. We believe that CloudBridge can be deployed for a variety of different brain imaging volumes and can significantly accelerate the pace of scientific discovery for neuroscientists. And we hope this tool can be valuable for all of you listening. Lastly, I'd like to thank my PI, our collaborators at Johns Hopkins and at Stanford for making this work possible. And again, here's a link to our code.
Hi, I'm Chris Xu from Applied Engineering Physics from Cornell. I represent Cornell NeuroNex Hub for large-scale activity recording. And my co-PIs are Joe Fetchel, Mercer Banju, Chris Schaefer, and Nilaya Pichi. It's a long title, but I hope you remember that what this hub does is to imaging deeper, wider, and faster in various animal species. And this is an overview of Cornell NeuroNex Hub. On the one side, we're developing techniques for imaging deep, wide, and fast. Of course, these areas are interrelated. We use those technologies developed to enable us to do impossible neuroscience research. Specifically, we're imaging adult zebrafish, imaging a fly through the cuticle, and imaging mouse brain and spinal cord. We're also using those neuroscience experiments to further inspire us developing new imaging techniques. As a hub, of course, we do technology development and also technology dissemination. And here's a brief overview of our accomplishments in the Cornell NeuroNex Hub. We have imaged adult zebrafish. This movie shows imaging adult zebrafish telecephalon areas from the top of the fish almost to the bottom of the telecephalon area. We're imaging the mouse brain spinal cord activity in a mouse running on a treadmill. We also can image a fly through intact cuticle and look at their brain activity. We can also imaging through intact and unthinned mouse skull. We can take movies over multiple days and see neuronal activity at a single cell resolved resolution. We've quantified the comparison between 2-photon and 3-photon imaging in terms of imaging GCamp. We have also developed a laser source that allows us to imaging the region of interest only. We call adaptive excitation source. For technology dissemination, we follow a simple strategy. First, we demonstrate convincing value for neuroscientists, and then we build an infrastructure to lower the adaptation of the technology we develop. And we also work with commercial vendors to make sure that our technology can be put into more biology-oriented research labs. And so far, about 40 to 50 labs have started 3-photon imaging on various animal species and organs, and also developing various different techniques, including, for example, head-mounted 3-photon microscope. And the 3-photon microscopy can also evolve beyond the brain into cancer biology and immunology, for example. And many institutions have adapted this technique, and some of them have already achieved quite good results. And upcoming results from the hub is we're looking into what is the depth limit for 3-photon microscopy, and how can we improve 3-photon signal strength. And those I will leave for the details for question sections.
Well, that was a fantastic series of talks. Thank you all for the effort that went into them. And there's quite a lot of activity going on in the Zoom chat. And I want to thank people for sharing their links and having these conversations on the side, as well as for some of the questions that have emerged. So I'm going to just start from the top. So there was a question in particular for Uri from Josh regarding how generally will these methods work out of the box? For example, would they work on sparse fluorescence data? Also, is the code available? And Uri has responded, but I'll let you unmute Uri to say if you want to have anything more about that.
I don't have more to say than what I typed in the chat, but I'm sure not everyone's reading the chat. We made all of the code as well as the training data completely publicly available to download off of 3dem.org. And in terms of the question about the applicability of the model to other stuff in fluorescence, it's a lot harder because the data is so much more sparse and there's so much more variability in what that's going to look like depending on the cell type, microscope, label, etc. But if you are imaging organelle trafficking in neurons, I imagine our model should work pretty well, but we haven't tested that. For the EM, it's pretty, as you saw, it works well for completely different microscopes, but it's all neural tissue. So if you try to apply our model to, let's say, muscle tissue, it's going to look pretty weird. It's probably not going to work. Yeah, but we've already done some new transfer tests where it's getting pretty good results with marmoset tissue, for example. It was prepared in a slightly different way. So it's pretty robust. With some additional transfer learning, it's going to be really robust. So we're excited about that.
Thank you, Uri. And there are several other questions to Uri along the way in this same vein. So I think we can look at the chat and his responses there. There's a question from Becca Pfeiffer and also from Terry Sanowski to Tom Bartol. The first question, Becca, maybe you want to go ahead and ask your question to Tom, if you're here.
Sure. It looks like he just answered. I was just asking whether or not line EM was able to detect sections potentially out of order and that sort of thing. Serial EM doesn't often have this problem, but serial TEM can occasionally have sections out of order.
Well, I answered it sort of in the chat, but I'll say it here on the Zoom. Art Wetzel's on board, so if I say something wrong, he can correct me. But as far as I know, there's lots of analytics that can be gathered from the output of Swift IR and can be used to detect out of order sections, upside down, even left, right, reverse, flip sections, damaged sections. We haven't yet added use of those analytics in our Alinum Swift GUI around Swift IR, but that's one of the things we plan to do in the future.
And Becca, if I can just follow up on Tom's response for a moment, because we've been using it regularly. There is a tool where I mean, you saw that this was a data set that had flaws in it. It had dirt on it, like typical, serial EM images and standard protocols that are not on block, but post-section stained. And Alinum Swift just ignores them. It just aligns right through them. It doesn't get distorted by it. It's really remarkable. And then Tom, I think one other question that came up is how much faster is Alinum Swift than other standard protocols out there? And maybe John Mendenhall can help to answer that question.
Well, so John has been using TrackEM2, including the elastic alignment capability of TrackEM2. While in the meantime, while we've been working on the GUI for Alinum, for Swift IR, our GUI is called Alinum Swift. And after we had it ready for use, he then compared the quality of the alignment and how long it took him to do it in TrackEM2 versus Alinum Swift. And he got a better quality, even though we don't have the elastic part working in the GUI yet. He got better quality than the TrackEM2, where TrackEM2 used the elastic alignment. And he got that quality in about 50 times faster than it took him to do it in TrackEM2. And that 50 times faster is maybe mostly the human intervention time. So as you saw, I was able to align that whole stack just by loading the stack in and clicking a button. And in the GUI, and we could even automate all that and just say, just from the command line, just say, here's my stack, go, right? And it could possibly even be even faster.
Okay, so we'll move on to some more questions here. Let's see, I'm trying to look to see if there's some. So there's been some communication about other data from primates. It looks like that was answered. A lot of questions to Tom. I'm trying to get some questions to the other people here. So here's a question from Linnaea to Bobby. Are you looking at within species sex differences?
So the short answer, which or maybe I said it privately, apologies, is that we would love to. We're not exactly sure what's the right places or the right ways to do it. But luckily, Linnaea said she would help. So I will defer the question back to her.
And let's see, I'm going to just open the floor to other people who would like to ask questions or to ask their own, because it's going to take me longer to read them all than for people to just raise your hand and ask your questions, whether or not they're in the chat. Hello.
I think Bobby's raising his hand.
Okay, Bobby.
Uri, I thought your talk was incredible. I was just curious for just because of time, you briefly mentioned that the segmentation improves after upscaling. Do you have a number? Does it get 10 times better? Not yet. Got it.
No, but we're working on that now. We're seeing if it can even improve training, let alone running inference on segmentation, because you have that additional info. So it's very qualitative right now. All I could say is that we do semi-automatic segmentation very often, and it was just so much faster and smoother than otherwise. But there's no way to time it when you have a human doing it in that way.
Okay, so we still have several minutes to go. I would like to encourage some of the junior people to either ask questions or put up some comments. And if you raise your blue hand, since there's 5 pages, I can't see your real hand usually. In the chat, we can be sure to get you. Or just start speaking and you will come up.
Hey, Uri. This is Brian.
Hi, Brian.
This is a question for Uri. So what semi-automated segmentation solutions are you using?
What are we using for segmentation? Is that what I heard?
So you noted semi-automated. Are you
Yeah,
making out of what, like watershed approaches, or how? I mean, what's your?
Well, for example, we often use Elastic for smaller scale projects, which is a machine learning random forest-based algorithm. But we're actually kind of shifting away from that, doing almost everything with deep learning now. And one project we're working on is looking at how super resolution improves the deep learning pipeline. And one thing I didn't get to talk about, because it's not even really out yet, but we have Arlo Sheridan, who was trained with Jan Fjunke at Janelia Farms, who developed a totally new deep learning method called shape descriptors, local shape descriptors. It's an assisted learning, deep learning method that seemed to be working even better. And what we were really nice, they developed it for neuronal segmentation, for putting together circuits. But we're finding that if you add in the super res pre-processing step, then it actually works for organelles as well. But that's all unpublished and being developed, and not quite ready for prime time yet. But hopefully we'll have some pre-trained models that are pretty robust out of the box to share with the community soon. Our PSSR, as I said before, is all available publicly for download. We always make the code completely available so that people can start working with it right away, and we'll be doing the same thing with everything else we work on with the segmentation pipeline.
Okay, there's a question from Tatiana Sharpie. Would you like to ask your own question, Tatiana?
Yes.
You muted yourself.
Yes. So my question was, what are the specific challenges of this clarity data? What are the features that work best for aligning it and so on? This was a question for Big Group.
Yeah, so typically the highest contrast features are the ones that are most useful for aligning in the registration. We do a set of pre-processing steps on the clarity to improve the contrast profile of the image so that we can register in an automated fashion. So high contrast features include cortical boundaries, things like dentate gyrus, some folds in the cerebellum, boundaries of obviously delineated brain regions. Yeah, does that answer your question a little bit?
Thank you. Yes, thank you.
Mm-hmm.
And Amy, I know you have a question for Silvio. Silvio, are you here? I think he wasn't able to be here.
I'm here.
There you are. I see you. Great. So there's a question from Amy. Amy, do you want to go ahead and read your question?
Sure. Sylvia, I was wondering, how do you actually measure the molecular components in that presynaptic terminal? It's amazing. What's the secret?
Very much hard work and very little intelligence. So the thing is the following. It's a combination of the following three ways. So first of all, we do a lot of immunostainings. And so we immunostain some 120 different proteins one at a time and analyze them with STED microscopy one at a time. And then what you do is you try to get the rough positioning at a resolution of about 40 to 50 nanometers for every one of these proteins separately versus the postsynaptic density and the membrane of the spine. So you see the membrane of the spine for every individual spine, and for every individual spine, you also see the postsynaptic density. So you get an idea of where your molecules are roughly organized. Then this type of organization, which in the end that we went for about a sum of about 50,000 spines that we analyzed by one by hand essentially, then you can translate this information for every one of the proteins into a 3D map made in electron microscopy, which was made by 3D EM reconstructions from the same type of cultures. And then we made a sort of a transfer map, voxel to voxel, to project the positions of the proteins from the fluorescence pictures, the average fluorescence pictures into the average 3D EM data. Then, of course, this is only I mean, it doesn't give you the copy numbers of the molecules. So to get the copy numbers, then you have to do quantitative biochemistry to get the copy numbers per cell. And then you have to do a lot of imaging at the cellular level to get the copy numbers per spine. You know, so it's that's conventional imaging combined with quantitative biochemistry. And eventually you reach the copy numbers per spine. So now you have the copy numbers. You have the 3D EM tells you where stuff is located in terms of volumes and everything. And then you have your fluorescence, your super resolution fluorescence to tell you roughly where the stuff should be located. By combining these three, then you get these kind of 3D movies.
Heroic. And can I ask, was that on cell cultures, you said?
This was all because the problem is the quantitative biochemistry. You should do it in something as simple as possible and as clean as possible. So we've done it on the types of cultures that were introduced by Banker that are not having any glia contamination. So you have like glia-free neuronal cultures. They allow you to do quite precise biochemical quantifications. Otherwise, it's much tougher.
Thank you so much.
But what we've done, sorry, maybe the answer is too long, but what we've done a couple of years ago, we've made a little technique which is able to convert from such analysis to convert, you know, if you image in parallel such cultures and then a brain slice, you can get a lot of parameters converted for the brain slice. So you can kind of convert this information into what you would expect in a let's say in a specific synapse in a specific brain area. So we have a little technique for this made a couple of years ago. So published a couple of years ago. So the hope is that from such data we'll be able to get more information also for different types of samples, more complex and more physiologically relevant samples.
Great, thank you so much.
Thank you so much for that conversation too. I see that Jaro Oria, you have a question in the chat. Would you like to ask your question and also to whom you were asking it? Are you with us?
Yeah, I'm here. I was just asking about the imaged mouse brain, about whether the stitching for those images was done automatically as part of the workflow, or whether that's like a separate step.
Was that question directed to who is that question?
That was directed to, where is he?
Bobby.
Yeah, Bobby, yes.
Okay.
Sorry, I didn't see it. So there was very little stitching done because the process is a tomographic process. So there is some stitching of moving the field of view from one field of view to another field of view. But very little stitching is done because it's an intact sample and not separated. There's some stitching done in frequency space.
So it was all imaged together, the entire.
Yes.
Oh wow. Okay.
And then Prasanna, I think you had a question. Would you like to pursue that?
Yes, I have a question to Chris Zhu.
Can you
I
reveal
noticed.
your video, Prasanna, please?
Yeah, can you hear me?
Can you reveal your video? We'd like to see you too.
Oh, for some reason the video is not starting up.
Okay, then let's
Yeah.
just go with the sound.
Yeah. So, yeah, my question is directed to Chris Zhu. I noticed that the delta F by F value in his presentation is very low for 2-photon. So, could you please comment why the numbers are very low? How do you interpret if the signal to noise ratio is really very low?
Okay, I'm glad you asked this question because it seemed like this session dominated by EM. Finally, the optics question. Yeah, in this case, we're actually doing a comparison between 2P and 3P. If you think about the delta F over F, delta F is generated from fluorescence from the neuron, and the F is the total fluorescence you gather from the animal. So when you go very deep, that's one of the issues with 2-photon imaging is that in addition to the fluorescence from the neuron, you also gather a lot of fluorescence from out of focus. So as you go deep enough, then the 2P delta F over F would deteriorate. And that's exactly why we're pursuing long wavelength 3-photon imaging to overcome that. So if you look at the 3-photon delta F over F, it's actually maintained from surface all the way down as deep as you want to go. But in 2P, the delta F over F goes very small once you go beyond, let's say, 600 microns deep. Does that make sense?
Yeah, that makes sense. Thanks.
Okay, thanks.
Chris, I'm sorry that you have EM envy.
Yeah, I'm learning a lot of EM these days, I guess.
All right, so we have a couple minutes left. Did I miss any burning questions from the chat that people want to just speak up, and then your window will become lit up?
I have a question for Kristen. So, Kristen, you assembled an amazingly large and diverse team of individuals doing incredible science, and I wonder what you focused on when thinking about designing this team to be able to achieve the goals that you have for your project.
So in the beginning, it was interesting because I started thinking about putting together a team, and literally the next day I received an inquiry from Silvio and Niels with a fairly well-established set of people that they wanted to include, and a totally different topic. And I said, I don't want to do that topic. The topic was aging, and I had tried to do aging synapses 3 times and failed miserably for a Pioneer Award, for various other things. I said, no way am I going to do aging. That's a topic that will die. So we immediately, in fact, they were thinking about, synaptic aging as the topic. And so immediately the German team and my team from our hub started to think about, well, what is a question that is really more central to the topic of connectomics across networks and across from molecular all the way to behavioral. And then I knew I really wanted to have a tool development team. So, it's interesting. I did very specifically look to have people from diverse backgrounds and to include as much as possible some gender diversity and diversity across cultures. And so that was very specifically thought about as we were doing it. But I must confess that the primary focus was to look for people who, you know, once we designed the 4 global topics, for people who would, you know, make unique contributions to each of those global topics, and also bring sort of different backgrounds and different tool knowledge. And I also, if you look at it too, I tried very hard to have really senior people who've been in the field for a long time. And I have for a very long time wanted to work under Mark Ellisman. So I was really thrilled when he said yes, that he would lead the subcellular team. He brings to us, you know, basically a very long history of exploring these issues. And then I had only just really begun to start working with Silvio in some of the work that he'd done, and Masa Kuijima in my lab had started to feed animals so that we can do some of the radiation detection drop and look at recently synthesized proteins. So, and of course, you know, Linnaea was a graduate student with me, but is a junior person who's in the team. So there was a combination of wanting to have balance in history and experience, and also right away to be building the team at the junior level. Uri Manor also is, you know, an amazing scientist, but relatively junior in the field. I mean, relative to me and Mark anyway, in terms of age. And so there was an effort to try to go from, you know, senior experienced people in each team, have some gender balance, and also to have some junior people in each team. So in fact, there was a real effort to do that. But I must say that it was primarily driven by the science and the question, with that secondary effort to have the diversity from all the different perspectives that I mentioned. Does that sort of answer your question? It was intentional. It wasn't unintentional. But at the same time, it was totally driven by the science.
I think it's a beautiful and honest answer that I really appreciate hearing from you.
Thank you.
As one of the leaders, female leaders in STEM, I and many of us look up to, so it's great to hear your perspective on those things.
I wish I had even more to choose from. And that's one of the things that I'm hoping this next generation is going to really give to us, and reaching out across the cultures and the generations and the genders to really bring in a lot more people into connectomics. I must say, the first year that I organized the connectomics meeting, I think we maybe had 3 women that were there as presenters. And even 10 years later, there were only 7. So this field really can use some infusion of some other perspectives. But we're lucky to have team members in all of our teams from across gender, across disciplines, and across levels of experience. So that has been a really a great joy. I just, all I can say is that we're going to dig deep and keep bringing up the next generation of people working in the field, I hope.
Linnaea, do you want to introduce the next session?
Kristen, maybe you allow me to say 2 words?
Yes, absolutely, Silvio, please.
Because I feel like you are very modest and you didn't really want to tell everything, but maybe I should just give the listeners a perspective from somebody who has seen you manage this whole consortium. And I should say I've managed a couple of these things before, you know, in Germany, and, you know, made a building, made a center, made another center. And I thought I know how these things are working. And then I started working with Kristen. And then it's a whole new perspective. I mean, to really coordinate people from 4 countries to get everything done. And you know, we had meetings. I was shocked, meetings with 20 people that have to present very short talks. and we end it on time by the second. I mean, I don't know how she does it, but I've never seen something like this before. I mean, this is the first time in a consortium that I'm like, I'm happy to be led by somebody else. I'm learning every day. I mean, this is something new. So honestly, I don't know how Kristen does it, but it's an amazing job. And I mean, it's been really for me a learning experience to just be part of a consortium organized in this fashion. So I think it's a very cool thing. It's not just about the science. It's also about learning how to do these things for the future. So yeah, thanks a lot, Kristen.
Thank you, Silvio. And I guess I have to say that I have a really wonderful project manager administrator in Anna Maria. And so anything I don't want to do, I just say, Anna Maria, will you take care of it and make it happen? And she does. In the same way, any thing that I don't really want to do from the point of view of organizing, I always have a Linnaea. I always say, if I can't do it, Linnaea will do it. So, and that has definitely happened. So now that we've had our kumbaya moment, I guess we can go back to Linnaea to open the next session.